Okay, is everybody there? Okay, now what I want you to do, let's put in our gene, A2ML1. Hit search. You'll see one gene comes up, macroglobulin. That's what it is. So let's click that gene. Again, remember at the beginning I told you about, you go in here and you try to get the criminal history. Well, this is like a database where you can get all that criminal history on this gene, right? I could look here. Here's its protein. Here's its structure. This is really cool. I love this. Has anybody been playing around with the AlphaFold? God, that's so cool. It's basically a way, we've done so many, protein, we've identified so many protein structures. What they've done is they basically created an algorithm you can give it any kind of protein sequence and it'll basically do the construction for you. It's pretty accurate as far as I can see. But again, you can go in here and you can see these molecules, right? Look at that hole in the middle. That's cool. In, out. A lot of times you'll see especially when you look at signaling molecules or you'll see a pore, it'll be like a perfect circle and there'll be a hole in the middle. You can actually see the active sites. What I also want to stress to you is that these genes aren't just two-dimensional genes, right? They make protein products. And these products have structure. And it's the structure that actually gives it its activity. So it is important to kind of pay attention to the structure. All right, hit your back browser. Disease-related gene. We can look at protein expression. It seems to be expressed in the epithelia, which makes sense for breast cancer. You can see where the RNA expression is. You can see basically different types. You can see if it's expressed in immune cells. So in this case, it is not detected in immune cells. A lot of times with some of these cancer targets, what you'll do is, you basically throw it in here and you'll see it it's really, a lot of the expression happens on the immune cells. If you see that, you're probably thinking, okay, this gene's involved in something, some alteration to the immune system that allows the cancer to continue. For these cancers to work, in order to grow, they have to hide. The immune system is always out there looking to take all these different cancers down. If it can hide, if it could somehow blind the cop on the immune cell, it has a better chance of proliferating. So a lot of times you'll see that. And whenever when I'm looking at a cancer target and I see that it has a strong immunological connection, that makes me think, okay, this is probably one of the mechanisms involved in why this thing is getting messed up. Go to cancer cell lines. We can take a look at some of the cancer cells. We can basically click on that. So again, this is cell lines. These are cell lines it's being transcribed in. A lot of head and neck cancer for sure. We do see it in breast cancer. I doubt this would probably play a role in skin cancer because then you can kind of look at these other different types. This is really cool too. This is where this thing is getting its own gang. So what it does is it's kind of clustered, basically the expression of it based on, things in its database. So this is the cluster it makes. We can analyze that as well. A lot about keratinization. Keratin plays a huge role, but again, you can go through here. You could see, all right, where's my breast cancer? Sure enough, definitely differential expression in lots of different types, cell lines associated with breast cancer. If I was interested in the brain, I could go there. I could go look at pathology. I could see basically pull up different studies. Hit up pathology real quick. I could basically get pictures of where it's located. I clicked on the breast cancer one. Not a whole lot of staining there. Go back. Ooh, I think they have single cell. Yeah. Click on the top. Click on single cell. This will tell you exactly like what individual cell lines that it's actually being expressed in. You go to breast cancer and show where like the different types of breast cancer tissue it's being expressed. In this case, C4, C20, what is that? Myoepithelial cells, it tends to be. C4, glandular cells maybe. Back. And let me see, where's the one that, I think it's, go to subcell. There's one where it has like a diagram and it actually has a, oh, here it is. So anything in green is where it shows up on the cell. Here's what I think is very interesting about this cancer is this gene product is being secreted. It's a protease inhibitor. Something associated with estrogen signaling. You know, if you have estrogen signaling, you can't have this protease. It's not being expressed out in the matrix. Then you could see that, you know, if it was in here, you would see like a bunch of staining within the nucleus. But again, you know, this is just kind of an example. And again, bookmark this page. The Human Protein Atlas is going to answer a lot of questions for you, especially on individual genes. To look at staining, I think, is really good about this. Also, if you want to look at single cell, you'll know exactly what cell types your gene is expressed in. I think this is a really good website where you can actually get a lot of good information on any particular gene or genes that you'd like.